Hi team, welcome to Controlling Microbial Growth in the Body, Part 3. This video we'll talk about antimicrobial drugs, drug tests, and the very scary antimicrobial resistance. So, drug efficacy tests. Efficacy just means means effectiveness. So, this cartoon, or this actually it's not a cartoon, it's a photograph, shows a plate. So here's an agar plate, and what they did is they created a lawn of bacteria. So they used a cell spreader, and they dipped their cell spreader into a culture of microbes, and then they just spread it back and forth and back and forth and back and forth and back until they created this lawn of bacteria. And then they took these paper discs that are soaked in an antibiotic, and they have a code name. So this is K30 that I just circled. Here's AM10. And so then you know what drug is on the disc, the white paper disc. And then you put the paper disc down, then you incubate the plate for however number of days you want to incubate. And you come back and you find that the drug has diffused out of the paper disc and killed all these bacteria within this range. Okay. And then what you can do is you can measure this diameter, or the killing zone, and you can see how effective your drug is. So it's when you measure the diameter of dead cells, so from here to here, the diameter, that's called the zone of inhibition. And it's a clear zone, no growth of bacteria, because remember the drug has diffused out of the paper disc and killed all the bacteria. So this zone of inhibition is determined by the effectiveness of the drug. So how effective is that drug in killing that microbe, and the ability of the drug to diffuse out of the paper disc. So that gets a little tricky, but I mean, think about it. If you made, let's say for this disc, your drug was really big. So it's a big old molecule, and it takes time to diffuse, so it didn't kill very many microbes in 2 days. Let's say in this one over here, GM10, drug GM10, let's say your molecule is really tiny, so it could diffuse really fast. So in the 2 days that you were incubating this, it could diffuse out and kill all these bacteria. So you kind of have to take a lot of things into account to decide how effective or efficacious your drug is. And then you can, once you have the zone of inhibition, you can use standard tables to compare how effective the drug is. This is called the Kirby-Bauer test, and we do this actual test when we are in the actual lab. More drug efficacy tests. This is called the MIC, or minimum inhibitory concentration test. And when you do this test, what you're trying to find is the smallest concentration of the drug that will inhibit growth. So let's say the drug costs $100 to make. You want the smallest concentration of that drug that will kill that will kill the microbe effectively. So let's look at the photograph. So it looks like it's called the drug's Tetra. Yes, and so here's the whole culture tube, and then when it looks like this, when it's kind of cloudy or turbid, that means the bacteria are growing well. When it's clear, like it is like in the tubes over here, this is all very clear, the bacteria have been killed. So what you can do, and then on the x-axis it shows the increasing concentration of the drug. So at 50, can't read I don't know what the denominator is, but this 50 micrograms per mil, probably, yeah, that's really high concentration of the drug, 50 micrograms per mil, killed the microbes. That's great, but check it out. 1.6 micrograms of the drug per mil also killed the bacteria effectively. So if you are a manufacturer, which one are you going to use? You're going to use this concentration because it's a lot. 1.6 micrograms per mil is a lot less than 50 micrograms per mil, and you can still kill the same amount of bacteria. More drug efficacy tests. You could use this E-strip. So again, can you see how this? I like this photograph because you can really see how the lawn of bacteria was made. So just going back and forth and back and forth. Probably used a sterile swab or something to really get a nice lawn of bacteria, of the bacteria of interest. And then you place this strip, this E-strip, this white thing right here. You place this strip that has a gradient of antibacteria, of antibiotic. So it doesn't have the units, but this is 256, pretend it's milligrams per mil, down to 0.016 milligrams per mil of the drug. And then you can see at these different concentrations, and the drug again will diffuse out of the strip, and then you can see how what concentration kills what amount of bacteria. So you can see this for 32 milligrams per mil, the zone of inhibition, or that diameter, is some number here, and then at 256 the diameter is a little bit bigger, so it kills more. So at that concentration of the drug, 256 milligrams per mil or micrograms per mil, for example, will kill more of the bacteria than 32 micrograms per mil. And then so you read the minimum inhibitory concentration directly from the strip. So the minimum concentration that will inhibit these bacteria, you could read maybe at 1 microgram per mil because it's still killing the bacteria. There's a minimum bactericidal concentration test. It determines the amount of drug required to kill, not just inhibit the pathogen. So in this cartoon, you have the concentration of the antibacterial drug at 8, 16, and 25. And this time they give you the units, micrograms per mil. And you have the microbe living, or mixed, the microbe mixed with that concentration of the drug. And then you plate the microbes that have been exposed to the drug on these plates and to look to see if they grow colonies. So at 8 micrograms per mil of your drug of interest, that didn't kill the bacteria. They're still growing in these colonies. At 16 and 25 micrograms per mil, there's no growth. So from a cost standpoint, you could use this drug at 16 micrograms per mil to kill your bacteria of interest. Okay, so let's talk about routes of administration of drugs. You guys probably know these because a lot of you have done, you volunteered as nursing aides. So routes of administration. You can give a drug topically. That means the drug is applied directly to the skin. So we come over here, we see topical. Looks like they're putting some cream on the skin here. You could do a PO route, that's orally. That's the simplest way to do it, I guess. And if the patient may not follow the instructions on these the PO route or oral route. So here in this cartoon, here's oral, here's an inhaler. Also, you could do an IM, or an intramuscular administration. That's here, so you give them a shot. And then IV over here. IV, intravenous administration. So you kind of have to continuously administer that, or the kidneys and liver will rapidly remove the drug. The physician has to consider how the drugs are distributed to the infected tissue. For example, if you have a drug and it's removed by the kidneys, that's not a good drug to use to treat a urinary tract infection. It's not so good for the heart. Sorry, this slide I've stopped a couple times. But anyway, this drug, metronidazole, oh, I bet that's how you say it, metronidazole, is effective on both bacteria and protozoa. So we have protozoa here and bacteria over here. So it's effective on both of those. So you would call this drug a broad spectrum drug. Tetracycline, so here, tetracycline is a broad spectrum drug, and it acts by inhibiting protein synthesis. And it's effective against gram-negative, gram-positive, chlamydia, rickettsias, but the side effect, it becomes incorporated into bones and the developing teeth of a fetus. So if you're a pregnant woman and you take tetracycline, this could happen, and it causes when the tetracyclines get incorporated in bones and developing teeth, causes malformation of the skull and this staining of the teeth. Some side effects of drugs could be allergies, could result in anaphylactic shock. So that's what this cartoon is showing. Anaphylactic shock just means you can't breathe. You could disrupt the normal flora in your intestine. So this is a cartoon of the large intestine. And if you open it up, you could see the overgrowth of normal flora, so you cause a superinfection. This is pseudomembranous colitis. So C. diff, Clostridium difficile in the colon, could overgrow and cause a superinfection called pseudomembranous colitis. Types of infections. So there's a lot of different ways to categorize infections. This is just one way. You could have a primary, secondary, or a superinfection. Primary infection, you get that directly from a specific pathogen. So for example, strep throat. Secondary infection results indirectly due to another pathogen that down the line will cause, for example, AIDS. And then a superinfection is when you have a secondary infection that occurs during antibiotic therapy for the primary infection. So initially you're treated for E. coli mediated urinary tract infection. So you treat for that. And then you get because of that, because you're treating the E. coli mediated urinary tract infection with the drug, you get a secondary infection by Enterococcus fecalis in the blood, and that's called a superinfection. So basically down here, it says a superinfection is a type of secondary infection that can develop when antibiotics kill much of the patient's normal or resident flora, and then one type of microbe will replace that normal flora, and you'll get very ill. Superinfection. So how do you develop resistance to an antimicrobial drug? So here's our plate, our agar plate, and we're growing these colonies. Each one of these is a colony. And this red thing right here, this red colony, is a drug-resistant mutant. So they're all the same types of bacteria. They're all growing in these colonies, but these red colonies are resistant to your drug. So then you expose the plate and the colonies to the drug, and look what happens. All the drug-sensitive cell colonies are killed. I don't know why they didn't kill that one, but anyway, and then the drug-resistant colonies survive. and then they grow here, and they'll grow these huge colonies over time. And then, I'm not really sure, most of the cells are now resistant. I'm not really sure why they put this guy growing, because initially it's supposed to be a drug-sensitive cell, and it should have killed it. But anyway, so in letter A, you have population with both drug-sensitive and resistant microbes. You expose the population to the drug, and it inhibits or kills the sensitive cells. Eventually, resistant cells are the majority in the population. So I guess maybe your drug of interest doesn't kill even these drug-sensitive cells. So they must be partially drug resistant, or they wouldn't survive. So when you do this, when you selectively kill sensitive cells and you allow the resistant cells to grow, you get these drug-resistant populations. Causes of the proliferation or increase of drug-resistant microbes kind of out in the world include subtherapeutic doses. So if you don't take your medicine, or they give you too low of a dose, you won't kill all of the cells, the microbes that you're trying to kill, and you'll develop these drug-resistant mutants or populations. And then also, if you have a superinfection, or if you're using a drug long-term, aka chronically using a drug, you can also cause an increase in these drug-resistant microbes. How do they become resistant to the drug? You can, when they're exposed to the drug, the bacteria could get new mutations on the genes in their chromosome. The bacteria could acquire plasmids from other bacteria, and the plasmids would have genes that would confer resistance. So for example, R plasmids, aka R factors, are pieces of DNA that confer or allow the bacteria that gets these genes to be resistant to bacteria. You can have resistance among bacteria, viruses, and protozoa. Initially, the resistant cells are usually in the minority in the population. So here in this first part of the cartoon, the drug-resistant cells are in a minority. The drug-sensitive cells are in a majority. But then when you expose them to the drug, that flips. So presence of the drug itself does not produce resistance. It selects for organisms that were already resistant. So when you give the drug here, these guys that survived in the red colonies, they already had some kind of mechanism to resist the drug. The drug didn't cause these bacteria to then develop a resistance to the drug. And also, patients don't develop resistance to the drug, microbes do. That looks like an important test question. The problem of resistance to antimicrobial drugs is a major problem for us. So for example, methicillin-resistant Staph aureus, MRSA, you probably heard of that, and then all these others. So how do you develop resistance? If you are a microbe, how do you develop resistance to antimicrobial drugs? You can do horizontal gene transfer. That's the source of antibiotic resistance between bacteria. And horizontal gene transfer can take three forms. Conjugation, so this part of the cartoon here, conjugation can take the form, horizontal gene transfer can take the form of transformation, and horizontal gene transfer can take the form of transduction. So let's look at transformation first. So here is, this looks like the prophage, or a virus that infects a bacterial cell. So this is the bacterial cell down here. So this is all bacterial cell. And then this, in transformation, you just have these short fragments of DNA, naked DNA. So they're not, in transformation, the DNA that will transform the bacteria is not in a capsid or an envelope. It's just naked. And it moves through the phospholipid bilayer of the bacteria and inserts itself into the DNA of the bacterial chromosome. In transduction, you use bacteriophage to transfer that piece of DNA that could confer resistance to a drug from one bacteria to another. And then in conjugation, so here's the bacteria up here. Here's the conjugation pilus. Remember, this is, and then what happens is this bacteria will insert a plasmid DNA into the recipient bacteria. This is not sexual reproduction, by the way. Some people are confused about this. This bacteria here is just sending a plasmid with a piece of DNA that will help the recipient bacteria become, or could help the recipient bacteria become resistant to drugs. In addition to horizontal gene transfer and these three types of horizontal gene transfer, you can also, if you are a bacteria, you can also encounter a mutation that would enable you to resist antibiotics. And I've shown this cartoon before. So here's one bacteria. He's got a piece of DNA. Here's a second bacteria. And he says, hey kid, want to be a superbug? Stick some of this DNA into your genome. Even penicillin won't be able to harm you. So this bacteria gives this bacteria a piece of DNA that will allow this guy to resist penicillin. So what it could be is this piece of DNA could code for a protein that is a pump that will pump the penicillin out of the bacteria. For example. Okay, so here is a cartoon of active penicillin. So it's kind of complicated. It's got this lactam ring in green, and it says here beta lactamase or penicillinase breaks this bond. And when beta lactamase or penicillinase breaks that bond, you have, so the bond's broken here, now your penicillin is inactive. So if you are a bacteria and you don't want penicillin to kill you, you could, luckily, have this enzyme, beta lactamase, that when you're exposed to the penicillin, you have beta lactamase, you break this lactam ring, and now penicillin doesn't kill you. Over 200 lactamases have been discovered. So penicillin's been around for a while, at least en masse since the 1940s, and bacteria have developed ways to overcome the killing effects of penicillin. Okay, so this is a cartoon. It says a couple of bacteria. It says this guy is saying, but Timmy, you have to eat your antibiotics or you'll never become a big and strong bacteria. So that's kind of, it's funny because, you know, we're developing antibacterial drugs or antibiotics to kill bacteria, and some of them are just taking those drugs in, and those drugs are just making them stronger, making them more resistant to any drug that we might make. So if you are a bacteria, you are resistant, you are slowing or even preventing the entry of a drug into your cell. Resistant cells, you may alter the target of the drug so the drug can't bind to it. So if I'm a resistant bacterial cell and I have some piece on my bacterial cell wall that the drug can bind to. It may be the case that I alter, I mean, I don't do it consciously, obviously, but the DNA and the protein could be in place that I put a mechanism on my cell wall that will alter the drug so that it can't bind to my cell wall, and then it can't kill me. If I'm a resistant cell, I could alter my metabolic chemistry that the drug is targeting. I could abandon the sensitive step in my metabolic chemistry that the drug is targeting altogether. I could overproduce certain enzyme molecules that inhibit the drug. Resistant cells, if I'm a resistant bacterial cell, I could also use a membrane pump that pumps the drug out. And if you are bacteria and you are in the multi-drug resistant strain of bacteria, oftentimes you have discovered a mechanism to pump the drugs out of you so they don't kill you. And here's a cartoon. Multi-drug resistance. Wow. This guy's pretty nasty looking microbe, but he's got a lot of tools that help him resist any drug over here, the antibiotic that we try to make to kill him. So if you are a microbe and you are multiply resistant, you could be resistant to all types of drugs. And this happens a lot in hospitals and nursing homes where lots of drugs are used, so the bacteria are exposed to them and they can develop resistance to the drugs. Some of the big problems in these places: Staphylococcus, Streptococcus, Enterococcus, Pseudomonas, Mycobacterium tuberculosis, Plasmodium, that's a protozoa that causes malaria. They're all big problems in hospitals and nursing homes. And then in addition to multiple resistance, if you are a microbe, you could develop cross resistance. So if I'm a microbe, I could be resistant to drug A, but that also may confer resistance to drug B for me. So, woohoo. All right, let's look at developing antiviral drugs. This is really interesting because we're trying to do that right now with coronavirus. So some actions of antiviral drugs. You could design a drug that would inhibit viral assembly. So this step here, so the virus has usurped the cell's machinery and has made all these protein parts of a virus, and then you assemble them into the virus bud. If you could design a drug that would inhibit the ability of these parts of the virus to assemble. You could inhibit viral replication. So the virus, here it's where the virus initially comes in. The virus has a lot of tricks to replicate its DNA, but you could inhibit that. You could also inhibit right here, before the virus even gets in, the attachment of the virus to the host cell. You could inhibit that with the drug. Antiviral drugs are usually more toxic than antibiotic drugs or drugs directed against bacteria. Okay, how do you retard resistance to antimicrobial drugs? How do we do that? How can we avert drug resistance in bacteria? We could use high enough concentrations of the drug for a long enough period of time to kill all sensitive cells and inhibit others, so then the body can take over and use its natural defenses. We could use different combinations of drugs, so that if we kill one pathogen resistant to one drug, they'll also be killed by another drug using combinations of drugs. And this just shows, again, here's our plate. So you can see there's a lot of bacteria here. Here's our antibiotic disc with this drug, amoxicillin, clavulanic acid, and here's another disc with aztreonam, I think. But anyway, So the two discs together can create this large zone of inhibition. You could also, in our effort as humans to slow down or retard the resistance of microbes to our antimicrobial drugs, we could also limit the use of antimicrobials to only necessary cases. That's what I think is a good idea. We could develop new variations of existing drugs, and we can also look for new drugs. So people are looking for new drugs in ocean sediments, marine organisms, in people's mouths. It's kind of cool. So they're looking for new drugs everywhere.